Hello everyone, welcome back to our channel. This is Edugenesis portal. Today we are going to discuss the cell, unit of life. And before starting that, we should know that what is cell theory. First of all, I need to tell you that cell, first of all, discovered by Robert Hooke, and the life cell was discovered by Anton von Leeuwenhoek. So this thing we know, we have already got in our earlier classes. So you see, cell theory. Now, cell theory is given by whom? Cell theory is given by Matthias Claden, and Theodor Schwann. You see, Matthias Claden, what he has done, he has studied different kinds of cells, okay, different kinds of cell, different tissues of cells, and he found that all cells have a unique membrane. Are you getting my point? A unique membrane, which is known as a plasma membrane, and all these things. And plant cell are made up of plant products. That means the endoplasmic reticulum and all these things, cellular organelles that is present in a cell. And Theodor Schwann, what he discovered is that he is a zoologist, and he discovered that plant cell and animal cell are different. They both have a plasma membrane, which is a selectable membrane. That Matthias Claden, what he said, in Theodor Schwann, we know that Matthias Claden said that plant cell are made up of cell products and the cellular organelles, and Schwann has said that animal cell have one unique layer along with plasma membrane. It has a what you can say cell wall, but plant cell don't have, animal cell don't have cell wall, and plant cell have cell wall. Okay, animal cell don't have cell wall, but plant cell have cell wall. Okay, so but both of them didn't say that from where the cell arises. The cell, from where the cell arises, it has been said by the Rudolf Virchow, another scientist, and the statement of the Scleden and Schwann, along with Rudolf Virchow, togetherly they produced a one theory. And that particular theory, in that particular theory, the findings of the Matthias Claden and Theodor Schwann are being added along with the findings of Rudolf Virchow. What Rudolf Virchow said is that the omnicellula cellula, that means the cell arises from the pre-existing cells. One cell will divide and it will give birth to another cell. So omnicellula cellula. Okay, so there are three statements in a cell theory, and what are those? All cell are made up of the cell and its byproducts, and all cell arises from pre-existing cells. And number three, all the organisms perform their functions with the help of the biochemical reactions performed inside a cell. Okay, and virus are exception to the cell theory, because you see virus are living and non-living. They either they are living or they are non-living, we can't say. Virus sometimes when they are found outside the host body, they cannot move, they don't move, they are crystalline structure, they behave like non-living structures. But when they will attack the host body, they behave as a they behave like a living structure. So that is why virus is either a living organism or non-living organism, we can't say it properly, so we have placed it in a different group. Okay, an overview of a cell. So we know that eukaryotic cell and prokaryotic cell are there. Eukaryotic cell have a definite nucleus, prokaryotic cell don't have membrane bound nucleus, and eukaryotic cell have a cytoplasm. Prokaryotic cell also have a cytoplasm, but eukaryotic cytoplasm has membrane bound organelles. No, eukaryotic cytoplasm has a membrane bound organelles, but prokaryotic cell don't have the membrane bound organelles. Membrane bound organelles like mitochondria, microbodies, vacuoles, lysosomes, Golgi apparatus, endoplasmic reticulum, all these are known as cellular organelles, and they are membrane bound structures. But these things are not found in prokaryotic cell, because prokaryotic cell are the primitive cell. Eukaryotic cell is a modified cell, and they have a membrane bound structure. That means these are present. Okay, we all know these things. And ribosomes are found in both the cells. Okay, but rhizomes are non-membrane bound organelles. That means ribosome don't have membrane. That is why it is found also in prokaryotes. Okay, so what is left, you see, in plant cell, we will in plant cell we don't find centriole, but centriole is found in the animal cell. Are you getting my cell? In animal cell we will get the centriole, which will help in the cell division or the separation of the chromosomes, but centrioles are not found in the plant cells. And plant cell have what you can say cell wall, and animal cell don't have cell wall. Okay, now you see, mycoplasma is the smallest cell. It's 0.3 micrometer in length, smallest cell, and the largest cell is the largest isolated cell is the egg of ostrich. Okay, and in our body the largest cell is, or the longest cell is nerve cell. You see, nerve cell, it is the longest cell. Human RBC, what is the size? 7 micrometer. Bacteria 3.5 micrometer. So these are the shape of the cell. Cell can be of different shape. You see, our RBC have the biconcave shape. You see, red blood cell WBC have amoeboid shape, that means indefinite structure. And you see, this is a columnar or the epithelial cell of our body. It is a longitudinal cells, means different shapes are there. You see, this is a nerve cell. Nerve cell are branched and it is quite long. And you see, this one is a tracheae, you can say. It is found in the plant cells. tracheids, elongated cells, and these are found in the leaves of the plants in mesophyll cells, round and oval. So what I'm trying to say is that different cells can be of different shapes in different type of organism as they perform different type of functions. Okay, now we will discuss prokaryotic cell, and as we know that prokaryotic cell don't have modified nucleus, and they don't have membrane bound organelles. The smallest cell is mycoplasma. It falls under the prokaryotic cell only. Blue green algae, bacteria, all will fall under the prokaryotic cell. PPLO means pleuropneumonia like organisms. It's very important. Pleuropneumonia like organisms, okay na? And you see prokaryotic cell divide much faster than the eukaryotic cells, and they vary greatly in their shape. Are you getting my point? That means in the members of the prokaryotic organisms will show different type of cells. They can be of the different type of shapes and structure of the cell. Okay, so and they divide much faster in compare with eukaryotic cell. So you see these are the structure of the bacteria. In bacteria itself, the cell can be of different types. You see this is a typical eukaryotic cell, and these are the prokaryotic cells. So you see bacteria, these are the bacteria. So what are the size? Some bacteria may have a spiralium at their ends. Some may be of circular. Suppose you can say vibrio, not vibrio actually, PPLO, that means coma shape. Vibrio means coma shaped bacteria, and it is a coccus type. Okay, streptococcus, which causes TB or the tuberculosis. It is of this type of shape, round like. Okay, this is a spiralium. You see their size. You see spiralium bacteria, typical bacteria 1.2 micron. PPLO is 0.3 micron. Viruses, this is the shape of the viruses. Viruses are even smaller than bacteria. Okay, so these are the different type of shapes of the cells of prokaryotic organisms. Now you see most prokaryotes have a cell wall. So most of the members of the prokaryotic cell, they have a cell wall. Okay, we know that animal cell don't have cell wall, plant cell have, but bacteria have. Most of the prokaryotes have cell wall, and they have cytoplasm, and there is no well defined nucleus. They don't have nucleus. Their nucleus is naked, we can say, as they are prokaryotic organisms, and genetic material is basically naked, not enveloped by a nuclear membrane. They have a dark region. It is not known as nucleus inside the cell. The dark region, generally we call it as a nucleus, but they don't have nucleus. This is not a nucleus. This is known as a nucleoid, and generally our nucleus have a nuclear membrane surrounding it. They don't have nuclear membrane, so that's why it is said that they have a naked DNA. Okay, in addition to the genomic DNA, the single chromosome circular DNA, the many bacteria have a small circular DNA outside the genomic DNA. So you see, suppose this is a bacteria. So suppose this is a nucleoid. Inside the nucleoid you will find a one circular long circular DNA or single chromosome. Are you getting my point? But along with that, they have an extra circular DNA. They have an extra circular DNA in their cytoplasm, and that smaller circular DNA, double-stranded circular DNA, it is known as a plasmid. Double-stranded circular DNA, it is known as a plasmid. The plasmid, now what is the function of the plasmid? Why they have a extra circular DNA? They will provide the phenotypic characters to such bacteria. Are you getting my point? That means the expression, morphological character, the characters which are visible outside. So this plasmid will provide the phenotypic characters to the bacteria, and another function it will do is that it will provide the resistance towards the antibiotics. That means the antibiotics, suppose if you develop disease, we take antibiotics. Antibiotics what they do, they kill the germs which has caused disease in our body. So how they kill the germs? Suppose antibacterial disease, and antibacterial antibiotic, how it will destroy the bacteria. Are you getting my point? So this plasmid will provide resistance from such kind of antibiotics. Okay, so okay, now cell envelope and the cell modification. Now cell envelope and cell modifications are very important. You see, suppose in a bacteria cell, you will find the three kinds of layer. The outermost layer is known as the glycocalyx, next cell wall, then plasma membrane. Are you getting my point? In a prokaryotic cell, outer cover is a glycocalyx, then it will be a cell wall, then it will be a plasma membrane. So let us discuss about the glycocalyx. You see, glycocalyx can be of the two types. Number one, slime layer. Number two, capsule. You see, capsule is a kind of hard structure, and slime layer is a loose. Are you getting my point? What is the function of slime layer? You see, which protects the cell from loss of water and the nutrients, so that the nutrients and the water that is present inside the bacteria, so that it cannot pass to the outside, so that it cannot go to outside. This slime layer will protect, means what can they can perform that kind of functions, that is they will prevent the water or nutrients to get outside the cell. Now another is a thick and tough covering, which is known as a capsule. And capsule and slime layer, they are made up of what? They are made up of polysaccharide. That means the units of carbohydrate. They are made up of carbohydrates, and little amount of proteins are also present there. The capsule is responsible for the giving gummy and sticky character. So you see, what's the function of the capsule? The capsule will responsible for the giving gummy and the sticky character to the cell. It allows the bacterium to hide from the host immune system. The capsule will provide the sticky appearance and gummy appearance to the bacterial cell outer covering, and it will help them to adhere. It will help them to attach, or it will provide the adhesion property. That means it can get attached to any substratum or any substance that it wants to. And this particular capsule will also provide the bacteria to get hide from the what you can say antibiotic maybe, or the things which can destroy them. They can hide from the immune system of the host. Are you getting my point? Now what is the function of the glycolytics and what they do? I have already told you. You see, the glycolytics is made up of polysaccharide macromolecules. They help in the adhesion. That means to attack. Are you getting my point? And due to the on the basis of the thickness, it can be of two types. One can be slime layer, another can be capsule. Okay. The next wall is the cell wall. Now, what does cell wall do? You see, the cell wall provides the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. Though the cell wall, they will not allow the organism to get burst out. Are you getting my point? They will provide the organism a definite shape. Are you getting? Now they will also provide a strong mechanical support to the organism, and this layer is very rigid due to one kind of compound which is found in them. And that means the cell wall of bacteria, suppose it is made up of one kind of very rigid structure which is known as a peptidoglycan. Peptidoglycan is itself made up of murin and a mucopeptide. And a number of antibiotic, example penicillin, inhibits cross-linking of peptidoglycan strands. Therefore, cell undergo lysis in the presence of these antibiotics. So you see, if antibiotic penicillin, I think you have heard about this name. So penicillin, how does it kill bacteria? It will destroy the cell wall of a bacteria. Then all the things present inside the cell, it will go outside, and the bacteria will eventually die of some purpose. So their cell wall is made up of peptidoglycan. So this penicillin will destroy the peptidoglycan components, murin and mucopeptide. So the security or the protection that the cell wall is providing, it will get lost. So ultimately the bacteria will die. So what is the function of cell wall and what it is made up of? We have already discussed. Next is plasma membrane. Okay. Before discussing plasma membrane, we should know that bacteria are classified into two groups. One is a gram positive and one is a gram negative. Okay, one is gram positive and another one is gram negative. Now you see, Christian Gram is the name of the scientist who have discovered one stain, and that's why this particular stain is also known as a gram staining. So that particular stain, if the bacteria takes, then it will be termed as a gram positive, and if it doesn't takes, then it is known as a gram negative. Are you getting my point? Stain is some kind of color pigment which if you provide to the cell, then they will show that particular color if you treat with them. Now if I apply the stain to a bacteria, and if the after washing, then if the bacteria still shows that particular color, then it will be a gram positive, and if it doesn't show, then it will be a gram negative. Are you getting my point? Now you see, as it is already written here. Okay, now we will discuss. Now the complete structure of the bacteria is given here. You can see this is the structure of a bacteria, detailed structure. You can see all the necessary plasma membrane and the cell envelope along with the cytoplasm and the things which are present in the cytoplasm. You see mesosome is there, lipid globule, glycogen, RNA, all these things we will discuss. The layer you can see. Okay, so now we will study the plasma membrane. So you see, this is the plasma membrane. The plasma membrane is a selectively permeable membrane. Plasma membrane is also known as a selectively permeable membrane. As I have said, the plasma membrane will not allow all the things to go inside or come outside. Are you getting my point? It will select that which things should go inside or which things should come outside. So plasma membrane is a selectively permeable membrane, and it is similar to in case of prokaryotes and eukaryotes. Okay, and one special structure you will see in a bacteria, that is, you see, plasma membrane will fold itself, and if important structure is made by the plasma membrane, which is known as you can see here, mesosome. This is a mesosome. You see plasma membrane, this is a plasma membrane. It will fold and it will produce one vesicle like structure which is known as a mesosome, and it will help in the many functions. For example, what are the functions? It will help in the cell formation, it will help in the cell division, cell wall formation, DNA duplication, distribution of daughter cell. So it will help in the cell division, cell DNA replication, that means the division of DNA, and the cell wall formation. It's a very important structure. Beside this, it will also help in the respiration. Are you getting my point? So you see, they also help in the respiration, secretion, increase the surface area of the plasma membrane, and enzymatic content. Are you getting my point? They can store the enzymatic content, they can increase the surface area of absorption. And mesosome is found in the gram positive bacteria, it is not found in the gram negative bacteria. And it is also found in the photosynthetic prokaryotes, that means we can perform photosynthesis. For example, cyanobacteria, purple bacteria, sulfur bacteria. There are other membranous extension to the cytoplasm are known as the chromatophores, which contain pigments. So plasma membrane not only form the mesosome. How the mesosome is formed? The mesosome is formed by folding of the plasma membrane, no? So it will not, the plasma membrane will not fold themselves and form mesosome, it will form some other structures also. Are you getting my point? There are many chromatophores also, that means there are many small vesicle will also formed by the plasma membrane, and they, suppose some of them are known as a chromatophores. Chrome, always remember, chrome means color, and chromatophore, phore means one sac like structure or bag like structure. So chromatophores or chromatobag contains what? Colors or pigments. Okay, now there are other structures also which is found in the cell, prokaryotic cell. You see, one cellular extensions, there are some cellular extensions, and those cellular extensions will produce some other structures also. For example, you see filaments, flagella. Are you getting my point? It is the folding of cell wall. You see, mesosome and this what you can say, mesosome and the other membrane bound structures are formed by whom? Plasma membrane, no? You see, invagination of the plasma membrane will form the mesosome, which will perform many functions. But invaginations or the foldings or the extensions, not invaginations actually, cell wall will extend itself and form some other structures. For example, flagella. Okay, now you can see the bacteria has formed flagella. These are the flagella. You can see this is the flagella. Are you getting my point? This is the flagella. So this, with the help of this flagella, the bacteria will move. So this flagella is made up of what? Who has made it? Cell wall. Okay, remember, cell wall. And the flagella is composed of the three parts: filaments, hooks, and basal body. Filament is the longest part and extend from the cell surface to the outside, and it is a hollow. It is a hollow structure, and it is made up of what? It is made up of special type of protein which is known as a flagellin. Flagella is made up of very important protein known as flagellin, and flagella is divided into three parts: filaments, hook, and basal body. I will show you how it looks. So suppose you see, this is a cell. Okay, this is a bacterial cell, a cell wall, and from here the flagella will originate. So you see, this portion is known as the basal body. This portion is known as a basal body. Here it is written basal body, and this portion is known as a hook. You see, this is a hook, and the longer portion, the longer portion is known as the filaments. Okay, now today up to this much, otherwise the video is already became very longer. So the next part of my video I will upload very soon. So if you like my video, then please like, share, and subscribe with your friends, and so that you can get the notification whenever I upload such kind of videos. Thank you.